So these slides are in many of my talks. You know, why did we even think about developing GBS? And really, it's very simple. And the simple answer is Ed Buckler said it would be a good idea if I worked on that, and I worked for him, so I did it. That's that's the simple answer. But really, it's because
that's basically the reason for most stuff that I do.
Yeah. So, but it was really all about, you know, agricultural work, plant breeding work, where you're going to do lots and lots of samples. You want a lot of markers, and you got to do it as cheaply as you can because the budgets just aren't that big, right? And so that was the goal, and also to address some other limitations. Of course, cost. We wanted high throughput, good replicability, and reducing the marker discovery bias that we see in some other systems. There'll be some extensive talk about that later. Okay, we've talked about this. The main features are reduced sample handling, very few PCR and purification steps, no DNA size fractionation apart from the PCR step, the barcoding system, this simultaneous marker discovery and genotyping, and it really scales well. I hope I've convinced you guys of all of those things. But why are people using it? One, it's actually pretty cheap, you know, compared to other marker systems. You get a lot of markers for a little bit of money. The molecular biology is really simple. I mean, how many of you people in here that work on the molecular biology? Okay. Does this look pretty easy? Yeah? Okay, so it's pretty easy. So you can do both this. I really like this feature, so I put it in like 6 slides. I don't know why. It's really robust. As you've seen, it works with a lot of different species, and you'll see a bit more of that in a minute. And as I mentioned earlier, it produces libraries that are ready to sequence on the Illumina platform without really any modification to the platform or the software that's running the sequencing machine or anything. So it's supposed to be easy from that perspective as well. Okay, keys to GBS data production. Again, high quality DNA. We will be stressing this again and again over this. It's just such a key portion of getting the molecular biology to work. So you need high quality, high molecular weight DNA that's free from contaminants that inhibit restriction enzyme digestion. So many of us have been, you know, working with PCR technologies for a long time. PCR is very, very forgiving. You don't need very good quality DNA to do PCR. This is a different story, right? If those people that have done AFLPs in the past, same kind of thing. And RADseq is the same way. Any technology where you're using restriction enzymes, you need to have good, clean DNA. There's no getting around it. You need accurate quantification. This is where I tell you what that means. Intercalating dyes, not spectroscopy. So if you think you're going to be able to do good work with the nanodrop random number generator, you won't. You won't. And if you've worked in molecular biology labs long enough, you know that that thing doesn't really quite do what you think it should, right? So don't use it. It won't give you good results. Accurate liquid handling, and this is what I was saying before, you know, if you get these 2 things right, but you mess this up, you're still in the same spot, right? So we typically, when we're doing high throughput work with this, it's robotics. It's good, accurate liquid handling robots that make this go. It's not to say that for smaller projects or smaller scale, you can't get away with doing hand pipetting, but if you're using a multi-channel, you want to make sure it's well calibrated. And who's calibrated their pipettes in the last 6 months? Okay, there's somebody, there we go. How about the last year? Yeah, a few people. Huh? Oh no, it's just the people. Now that's cheating. You know, it should be only the people that actually use the tool. That's. But you see what I'm getting at, right? You see what I'm getting at. This is really it's really key. Okay, the titration step, getting that right is important. Was that step reasonably clear to you guys? The titration step, where you're trying to get the right balance between the adapters and the genomic DNA? Okay.
Rob,
yes?
how long would that titration sort of process take?
Less than a day. Yeah. Yeah. So yeah, the question was how long would the titration step process take? Less than a day. Well, if it works the first time. Right? Yeah.
Depends if a bioinformaticist is doing it or a
Yeah.
molecular
Yeah, I'm not advocating, by the way, thanks for that, Jeff, because I'm not advocating that you all swap roles in this whole process. It's probably, you know, I've seen bioinformaticians in the lab, some, you know, a little scary. I've seen molecular biologists try to do bioinformatics too. It can be pretty scary. So this is why I want you guys to work together, right? So this one, number 5, I want to explain a little bit. So, you know, I've been harping on this idea that we want to make sure that the samples are matched, the genotypes are matched up to the appropriate sample. And a lot of what we're doing in this method is actually building in ways to check for mistakes, right? And I completely advocate for that in lots of different ways, as I think you've heard them this morning, right? This one here is really, really helpful. So, you know, you've got a 96 well plate. If you put 96 samples and 96 barcodes in there, and somewhere in the lab it got turned, you'll never know, will you? You'll never know. And that's a problem. You want to be able to catch these things. So by putting a random blank in each plate, and Katie, what's your comment on the random blank?
Please don't all put your blanks in H12 or A1.
Okay, that's not random.
Thank you.
Okay?
A blank in a random position.
That's
but
right.
Not H1 or
Yeah.
H12 or A1.
To give you some idea of how important this is, I was working on a project where there were multiple collaborative submitters of different plates, and because we wanted this to be correct over the entire project, we actually assigned, we made plate names that were made we had a naming convention for each plate. The name itself included the ID of the person who was submitting the plate as well as the placement of the blank. And we went through blank placements A1 to H12 before we repeated. So we wanted to actually cover all of that. And then I also wanted to make sure that people were actually putting their samples in the wells that they were supposed to be in. And not only did we have a blank on the plate, but we had 2 controls where we had them submit, in this case it was mapping populations, we had them submit maternal and paternal samples twice on each plate. And so what we ended up doing is we ordered the physical plates ourselves, took markers, and we colored black on the bottom of the well where the blank was supposed to be, and yellow and blue where the parental DNA was supposed to be, because it is very important that you get this right. And I can tell when people got it wrong, because if you, and this was with plant tissue, they were submitting plant tissue and the DNA extraction was done centralized. And we kept getting DNA in some blank wells, and it was isolated to one single lab, and we tried to figure out what was going on. Well, it turns out they had an absent-minded technician who kept putting leaf tissue into the blanks and then taking it out and moving it. It turns out you can actually get DNA from that, and so it kept flipping our red flags for quality control before we figured out what was going on. And so it's very low level, and sometimes this happens, and you can expect a low level of contamination in that case.
Yeah.
So
what?
Can you define a blank? The blank is everything in it except the DNA.
That's right.
So you still do your
You still put everything else in, but no DNA. And what this example that Katie has just given tells you that you need to do this blank at the collection stage, right? From the very beginning, you need to do this. And it goes along with the general philosophy I have of not moving things around much, right? And if you don't put something in there and then take it out, just don't put it in. You know?
in that example is with leaf tissue, we could still detect it. If you pipette a sample of DNA and then pipette it out, that's not going to be a low level of contamination.
That's going to be massive, okay? And from what Katie didn't expressly say this, but I'm sure you could infer that these things are checked in the bioinformatics pipeline, right? We look for them. So this is something that's really, really key. You want to catch mistakes. So, you know, I mean, we all make mistakes. Things happen. You know, that's just the way life is. But if we build careful systems to catch those mistakes, we're just going to be a lot better off in our science, right? So that's just crucial.
And if you make a mistake, don't try and hide it, because it's going to come out. Just fess up and go back a step when it's not so expensive as having to repeat your entire experiment.
You know, one of the most expensive parts of this whole thing is the sequencing, right? And if you're in the lab and you're doing library construction and you know you've made a mistake, stop.
Or you think you probably made a mistake.
Or if you even think you've made a mistake, stop. Go back a step. You know, do that, you know, repeat that step so that you're confident that it's right before you go spend the money on sequencing, and then somebody like Katie says, oh, this is wrong, you know? And then all that data has to get chucked because what are you going to do with it, right? So, and this last one, I always debate about whether I should leave it in or not, but so I was really careful when I wrote this protocol and developed the protocol. I could have part numbers and which companies, and you know, those are things that you don't often see. I know I'm sure a lot of you that work in the lab get very frustrated by these protocols that seem to have a missing secret ingredient somewhere. I just loathe that, so I did my best to just really spell it all out. This is what I did, and this is what will work, right? And I often have gotten questions over time about people wanting me to troubleshoot their protocols, and it usually takes 3 or 4 rounds of back and forth before I find out, oh no, actually, I thought you meant this, so I did something else. Or I did something else altogether. And I'm like, that's not my protocol. I can't, you know, you gotta tell me these things. It's very hard to troubleshoot something when you expect it's one thing and it's not. But this protocol works pretty well and pretty consistently, and if you follow it. Okay. This is the GBS applied to many species at the Institute for Genomic Diversity.
Pretty pictures?
Pictures. Okay, so we've got, these are slides that Katie made. Many of you have seen them before in my talks. You want to talk here? Or?
No, they're really just pretty pictures.
Okay, yeah. No,
The point being that we've done this on a lot of species now. And these are actually quite outdated at this point.
Yeah, I mean, there could be many, many more. But the point is that the Institute for Genomic Diversity has worked with all the species that I'm going to show in these slides and many, many more. So those are a bunch of grasses. Here's some vegetables.
Vegetables are interesting because some of the vegetable projects we worked on were particularly interesting because the question, how many SNPs will I get? Well, for some of these species, and in particular mapping populations that were narrow crosses, the answer was very, very few comparatively. And you know, I released some data and I thought, oh, these people are going to hate me. They only have a couple hundred good markers. And they were thrilled because it was better than the marker system they were currently using. So, yeah.
Yes, sir.
So do you even run into issues from interpods?
I haven't done
Do you
any.
ever run into issues from interpodic microorganisms?
I've got some experiments planned to actually try to do, so the question was, do we ever run into problems with endophytic organisms? And actually, we've got some experiments planned to see if we can do genotyping of ryegrass and antiphyte at the same time. Okay, so we're actually working on that because that would be really cool, right?
That comes down to the analysis.
And it comes down to sampling as well.
If you're aligning to the reference genome and the reference genome doesn't have any endophyte DNA in it, which it might, I don't know, then you'll get rid of it there. But you asked about wheat. Yeah, there's a guy at Kansas State, Jesse Poland, who did a sort of next little modification of Rob's protocol.
And Jesse's on our
paper. If you
paper,
follow
so
his protocol, it'll work too.
Yeah, it'll work too. If you follow his, it'll work too.
PSD1 and another cutter, and he uses that on wheat, so he's done a lot of GBS in wheat. Pardon me?
The density of markers then?
I think he gets wheat's so big, I think he gets similar results with PST1 and another 4-base cutter than compared to us using APK1, which is a 5-base cutter with wobble.
Yep. Okay, I want to push along here a little bit because it's lunchtime, or getting there. Yeah, here's a bunch of other things like cotton and chickpeas, sunflower, hops, which is one of the things many of us around here like. Tree species of varying sorts, fish, and even some things that our friends from the South Island might be interested in, like cows and so forth. And I know that they're doing sheep down there already, so. you want to say a few words about the strengths and weaknesses at this stage, or do we want
Yeah.
to save it for later?
Given that it's lunch, and I think that we're gonna continuously talk about strengths and weaknesses in this workshop, all I wanna say is that there definitely are strengths and weaknesses, and we're gonna talk about usage cases, which is important because not every case is going to be the same. We can show you some of what we've done and some of the things that have been strengths and weaknesses in those cases, but you'll all have to think about your project independently and figure out how this plugs into what you're doing.